This talk will be presented by Dr. Mengbin Chen, who is a postdoc in my lab. Mengbin is a fantastic scientist. She received her PhD from University of Pennsylvania, working with Professor David Christensen on the structures and functions of sesquiterpene synthesis. And she joined my lab more than 3 years ago. It's really lucky to have her in the lab. She has a broad set of interests in natural products in general. She's working on genome mining of new natural products from different filamentous fungi for both healthcare and agricultural applications. And during that process, she became interested in finding new enzymes in those biosynthetic pathways that can catalyze unusual reactions and potentially then use these in biocatalytic applications. So today, she will talk about a recent work that just completed on the discovery of a new type of PLP-dependent enzymes that form CC bonds from a biosynthetic pathway and its application to be to synthesize different chemicals. Take it away, Mengbin.
Okay. Thank you, Professor Tian, for the generous introduction. And I also want to thank the organizing committee for this great opportunity to present our work. So my talk today will be discovery and biocatalytic application of PLP-dependent CC bond forming enzymes. So non-canonical amino acids, including pipecolates, are important synthons in medicinal chemistry. We see them in some of the most famous natural products, such as rapamycin and treprostin. The incorporation of pipecolates can actually enforce a biologically relevant conformation of this macrocycle. Furthermore, they are also used in synthetic drugs for target recognition and binding. The chemical synthesis of pipecolates often require multiple steps of protection and deprotection steps and auxiliary reagents. Nature, on the other hand, has a relatively streamlined fashion to make L-pipecolate. L-pipecolate is often derived from L-lysine by deamination at the terminal amino position or the alpha amino position. It will give rise to an imine species, which will then further reduced by the imine reductase that gives rise to L-pipecolate. While L-pipecolate is quite commonly seen in nature, substituted pipecolates are rare. However, they are actually a keen target for medicinal chemists to pursue after. So we thought if we can find enzymes that can make substituted pipecolates, we can actually develop them into biocatalysts to further diversify the drug candidates that have pipecolates as a structural motif. So if we look, if the substituted pipecolates follow the same biosynthetic logic as the biosynthesis of unsubstituted pipecolates, they should be derived from this imine species. And by counting the number of carbon atoms in this imine, we know that they cannot be derived from proteinogenic amino acids directly. This indicates that there has to involve a CC bond formation event for the generation of this imine precursors. So the talk that I will be so the question that we wanted to try to answer today is that can we actually find enzymes that catalyze this CC bond formation event, and can we find a stereospecific reductase to reduce this imine? So one of the natural products that really caught our eye is prenolated indole alkaloid citrinodine. By retrobiosynthetic analysis, what we have noticed that one of its amino acid building block is L-tryptophan, while the other is quite exciting here, which is 2S6S6 methyl pipecolate. Again, if it follows the same biosynthetic logic as L-pipecolate, it should be derived from this imine species, which can be forged by primary metabolites, which we don't yet know about. long story short, we were able to determine the genes that are necessary for the formation of this 6-methyl pipecolate. So there are 2 genes that are absolutely necessary. One is a PLP-dependent enzyme, and the other is a short-chain dehydrogenase. So what we have noticed that if we add the 3rd gene in the heterologous host system that we use to reconstitute the pathway, which is a HMG-CoA lyase-like enzyme, the yield of compound 1 is actually increased by almost 2-fold. So the HMG-CoA lyase gene shares a very high sequence identity with the genomic copy. So we thought that the increase in yield could indicate that this copy actually provides the precursor to the 6-methyl pipecolate. And in this case, it would likely to be the nucleophile acetoacetate. So to prove that acetoacetate is indeed the precursor to the amino acid we're interested in, we used the C13 labeled ethyl acetoacetate and fed it into a heterologous system that's expressing the 2 genes that are essential for the formation of compound 1 here. So we were able to observe a +2 M/Z value, indicating that the 2 C13 labeled atoms were indeed incorporated. And we were also able to characterize the C13 labeled compound by NMR, and we showed that the while one of the C13 labeled atom is incorporated at 5 position, the other is at the 6-methyl position. And this also indicates that the new CC bond forms between 4 position and the 5 position here. So now we know that there are 2 genes that are essential for the formation while 1 is SDR. This indicates that the compound 1 here is indeed derived from an imine species, and the one of the building blocks of the imine species is acetoacetate. This indicates that the amino acid donor would be a good would have a good leaving group at its gamma position. So we purified this enzyme from coli, and we show that this is indeed a PLP-dependent enzyme by having the characteristic absorbance of 420 nanometer corresponding to the external albumin species. We also tested a bunch of amino acids that are in primary metabolites, and we showed that O-acetylhomoserine is the best amino donor here to form the CC bond. We also showed that the CNDE is indeed an imine reductase that catalyzes the stereospecific reduction of this imine species. We also characterize the steady state kinetics of CNDF towards 2 substrates, and the parameters are comparable with other PLP-dependent enzymes that have been discovered in secondary metabolism. So we now know that the CNDF is a CC bond forming enzyme. We wanted to know if it can be applied as a biocatalyst. Before doing that, we actually need to assess its substrate scope. So instead of using beta keto acids as the nucleophile, we decided to use beta keto esters as substitutes because they are more commercially accessible and they are more stable, so we don't have to worry about the decarboxylation step in the presence of primary amines. So the idea here is that we wanted to vary the substituent at R1 position and also R2 position. So if we increase the size of the substituent at R1 position from methyl to isopropyl to tertiary butyl and to cyclobutyl, we notice that the biggest substituent that can be accommodated is the cyclobutyl substituent. If you look at the bottom row, anything bigger than the cyclobutyl substituents such as cyclopentyl or the benzyl substituent cannot be accommodated at all. In addition to that, we noticed that both 5 and 6 position substituents can be accommodated if we use this beta keto ester as one of the substrates. We also tried out a bunch of fluorinated acetoacetate keto esters, and we were very glad to see that they can be fully converted. However, if the substituent contains a species that is likely to be protonated under the conditions that we tested, they cannot be converted by CNDF at all. Halogenated stereoxopropanoic esters cannot be converted either. So it seems that the CNDF has a relatively relaxed substrate scope, and we wanted to couple it with the imine reductase to see that if we can biosynthesize some L-pipe cholates with substituents at the 6th position. Because we wanted to start with something that's really cheap, so we decided to use beta keto esters, and we also wanted to take advantage of the lipases. Although we were not able to do this in vitro, we decided to use our heterologous system, which is a prolific source of promiscuous lipases and esterases. So the idea here is that if we fed the heterologous system with the beta keto esters, the lipase will just hydrolyze the ester group and generates the beta keto acids in situ. CNDF will take the beta keto acids and forge the imine species, which will then be then reduced by the imine reductase. So it turned out, so this worked out nicely. We were able to obtain a bunch of 6 substituted pipe cholates. You may have noticed that for compounds 19 and 20, we didn't really specify the stereochemistry at 5 position here. And this is likely due to the scrambling of the stereochemistry at 5 position at during the decarboxylation step. We think that the imine reductase, while it has a very nice selectivity over 6th position, it doesn't really have a preference over the over 5 position. So now the question really boils down to if we can find a system that has better selectivity at 5 position. So when my coworker was studying another biosynthetic pathway of mycotoxin fusaric acid, we found a similar PLP-dependent enzyme that uses O-acetylhomoserine and hexanol to generate the imine precursor. We thought if we could pair this PLP-dependent enzyme with a different reducing reagent or a different imine reductase, we may be able to synthesize 5 substituted pipe cholates. So indeed, we were able to do that. So as you can see here, if we use chemical reduction, the DR value is modest here, favoring the trans diastereoisomer. However, if we have an endogenous imine reductase from the heterologous system, you can see that the DR pattern is completely reversed, favoring the cis diastereoisomer. So we are currently in the process of identifying this powerful imine reductase. In addition to that, we can also use the PLP-dependent enzyme FUB7 to construct pipe cholates with a quaternary carbon center at the 5 position. It seems that this has very promising biocatalytic potential, so we are currently exploring its full substrate scope. So for the talk, I have shown you two PLP-dependent enzymes, and that can form CC bond, that can form new CC bond at the gamma position. So if we couple these 2 PLP-dependent enzymes with different imine reductases that presumably have different stereoselectivity, we will be able to obtain different stereoisomers of pipe cholates. So with that, I would like to thank my advisor, Professor Chen, for all of the advice and support that he has given me when I was working on this project. And I also wanted to thank my coworker Yang He for his efforts on FUBE7 and my undergrad mentee on FUBE7 on CNDF, and also other lab members. And the funding support is provided by NIH. With that, I'll take any questions that you have.
Okay, thanks very much, Mengbin. That's a fantastic talk. Can I just hand over to Andrew for the questions?
Great, yeah. Okay, so can SYND-F be used as an isolated purified biocatalyst working on soluble substrates with GL titer?
Okay, so yes, we definitely can purify the CNDF, and we also tested, we can actually have small scale, large scale in vitro reaction to produce milligram scale or at least 10 milligram of the pipe collates, but I'm not sure if we can further increase the final yields of the compound that we're interested in, because what we do have noticed is that the CNDF will be slowly killed because of the because of the nature of this PLP-dependent enzyme. So the O-acetylhomoserine will go into a shunt pathway, which instead of forming the CC bond, it will be deaminated and form the alpha ketobutyrate. So we think this is one way that really diverges the pathway from producing the compound that we're interested in. So I think there is still some way to go for the for large scale production, and it would require some optimization and protein engineering efforts of this enzyme.
Mm-hmm. Great. We have another one here from Roberto Chica. How reactive are the beta keto esters versus the beta keto acids as substrates?
Yes, so I didn't show the data here, but we did the steady state kinetics on the beta keto esters. So for ethyl acetoacetate, which has an extra ethyl ester group, the kinetics are comparable.
Great. There's another one here. Are there any efforts in engineering CNDF to accept other non-natural substrates? Has the crystal structure of CNDF been resolved?
We just started working on the crystal structure of CNDF. So when we actually found that, okay, FUBE7 can take in aldehyde as the nucleophile, we thought that maybe CNDF can do a similar reaction because aldehyde essentially goes through the enolate for the CC bond formation. However, we try to use other types of enolates. It doesn't really work. So I think CNDF has a relative strict selectivity over the pKa of the C alpha proton, because beta keto acids are relatively the proton, the C alpha proton, is really acidic. So the requirement is that you really have to have a highly activated C alpha position, which is maybe just mono substituted.